This is the Chapter 24 activity video for the digestive system. So Part 1, Alcohol and the Digestive System, Hospitalization One. Vincent Miller, a 62-year-old accountant, has had a, quote, drinking problem throughout most of his adult life. He drinks about a half case of beer each day. He has lost several jobs over the years for drinking at the workplace or showing up for work drunk. He lost his driver's license for drunk driving, and his drinking has placed a considerable strain on his marriage. He has been hospitalized on several occasions over the years. So let's take a look at Vincent's past medical records. Hospitalization One. Vincent was hospitalized at age 32 with a complaint of vomiting up blood after a drinking binge that lasted 7 days and was marked by excessive and repeated vomiting episodes. The vomit was bright red. The diagnosis was a tear in the mucosa at the gastroesophageal junction, which is where you find the lower esophageal or cardiac sphincter, caused by repeated vomiting. Question 1. Describe the 4 layers of the digestive tract lining. Which layer was injured in Vincent's case? So I'm going to put a picture up here so we can reference it. So we're going to start with the innermost layer, and the innermost layer is called the mucosa. So the mucosa contains a mucous epithelium and then underlying connective tissue called the lamina propria, and this is areolar connective tissue. The epithelium can be simple or stratified. So in areas of the digestive tract where there is not a lot of abrasion, you will have simple columnar epithelium. And in places where you do have a lot of abrasion, you will have stratified squamous epithelium. In some places, like in the small intestine, the mucosa is folded into these complex folds called circular folds or plicae circularis. The lamina propria contains extensive blood vessels, nerves, and intestinal glands. And the epithelium can contain mucous cells and enteroendocrine cells. Under the lamina propria, there is a thin sheet of smooth muscle called the muscularis mucosae. So, moving outward above the mucosa would be the submucosa. And this is made up of dense irregular connective tissue with numerous blood vessels and lymphatic vessels. It can contain exocrine glands in some regions, like is shown here in this picture. This would be a little exocrine gland. And it also contains the submucosal plexus, which is a layer of nerves that plays a role in the enteric nervous system. Above the submucosa, you would have the muscular layer. In this layer, you have smooth muscle that is arranged in 2 primary directions. There is longitudinal muscle, which is lengthwise to the tract, and then circular muscle, which goes around in a circle around the tract. And then in the stomach, there's a 3rd layer called an oblique muscle layer. The muscular layer also contains a network of nerves called the myenteric plexus, which is also part of the enteric nervous system. And then above the muscular layer, you have the outermost layer, which is called the serosa, and this is the same thing as the visceral peritoneum. So to answer the second question, in Vincent's case, the mucosa was torn, which would cause bleeding from the ruptured blood vessels in the lamina propria. Severe bleeding could also indicate that a tear extends into the submucosa, where you have additional blood vessels. Question number 2. Why was the blood bright red rather than the color of coffee grounds? So when blood is spent a significant time in the stomach, the acid in the stomach oxidizes the iron in the hemoglobin and turns it into a dark brownish black color. Therefore, blood that has been in the stomach for a while will appear dark and clumpy like coffee grounds. When bright red blood is vomited, it indicates more acute upper gastrointestinal bleeding to the extent that the blood has not yet spent much time in the stomach acidic environment. So here is a little guide showing the different colors of vomit. So if it's red or pink, then that means that there is actual blood, fresh blood that is newly entering the gastrointestinal tract, whereas either brown vomit or black vomit could indicate that the blood has been there for a while, or it's a slower bleed. Number 3, describe the vomiting reflex and what controls it. Why might severe vomiting result in a tear to the mucosa? So the vomiting reflex center is located in the medulla oblongata. And here's a little pathway illustration. I'm not going to go over every step in this pathway, but it does say some of the triggers that can lead you to vomiting. But when you have a stimulus that comes into the vomiting center, that is going to be in the medulla oblongata. And then the reflex is coordinated through several different cranial nerves. So as part of the vomiting reflex, the pyloric sphincter, which is the sphincter between the stomach and the duodenum, is going to relax. And then both the lower esophageal sphincter and the upper esophageal sphincter will also relax. And then reverse peristaltic waves, which in this image they're calling anti-peristaltic waves, will push the contents of the duodenum and the stomach up into the esophagus and pharynx and then out of the mouth. There is also a signal to contract the abdominal muscles very forcefully, and this forceful contraction of the abdominal muscles will increase the intra-abdominal pressure, which will also help to push the material upwards. So severe vomiting can result in what is called a Mallory-Weiss tear at the junction of the stomach and the esophagus, and it can extend into both the mucosa and submucosa. So Mallory-Weiss tears can be caused by a combination of increased pressure that is pushing out on the esophageal walls from the forceful contraction of the abdominal muscles and the reverse peristaltic waves, and you've got weakening of the esophageal wall by the repeated exposure to stomach acid because of a severe bout of vomiting. So as mentioned, this is called a Mallory-Weiss tear, and it's in the mucosal layer at the junction of the esophagus and the stomach. These can also be called gastroesophageal lacerations or gastroesophageal laceration syndrome. And Mallory-Weiss tears are often associated with alcoholism or bulimia, which is a type of eating disorder where people eat a lot of food and then make themselves throw it back up. But it can be caused by any condition that causes repeated violent vomiting and retching. So even a severe bout of food poisoning can cause this as well. If the bleeding doesn't stop on its own, the tears can be cauterized during an endoscopy procedure. So we're still in part 1, but now we're going to look at hospitalization number 2. So at age 36, Vincent was hospitalized again, this time with complaints of abdominal pain in the upper epigastric region, which is just below the xiphoid process of the sternum. And he was vomiting blood that looked like coffee grounds. He also complained of heartburn, which was partially relieved with antacids. A diagnosis of upper GI bleed due to gastritis and reflux esophagitis is noted in his chart. So number 4, explain how the stomach lining is normally protected from its own acid. What effect does alcohol have on these defense mechanisms? So the stomach produces a thick carpet of alkaline mucus to protect itself from the acids and enzymes of the stomach secretions. So here is an image to give you an idea of what this looks like. So you've got the cells that line the stomach. So as part of the epithelial layer, there are mucus cells in between the other epithelial cells. And the mucus cells secrete an alkaline mucus, so they secrete bicarbonate ions. And so they keep this layer of mucus right above the lining that has a pH of around 7, which is close to neutral. Whereas in the stomach lumen itself, where you have the acid, the pH can fall as low as 2. So the mucus layer provides like a buffer region between the acidic interior of the stomach and the cells that make up the lining. In addition to the layer of protective mucus, the epithelial cells are bound together with tight junctions to prevent acids and enzymes from leaking through to the interstitial area. Epithelial cells have a rough life, particularly in the stomach, and they are constantly being regenerated in the gastric pits. So a typical epithelial cell in the stomach lining only has a 3 to 7 day lifespan. So in the case of alcohol, alcohol is lipid soluble, so it is able to actually diffuse through the mucus layer and even penetrate into the epithelial cells. So it can irritate and damage the epithelial cells, leading to inflammation of the mucosa, which is a condition called gastritis. So here is a picture of a healthy stomach with a healthy mucosa, and then during gastritis you have inflammation of the lining, which is going to affect the mucosa inside the stomach. So chronic gastritis from chronic drinking can result in less mucus production and weakened defenses against the stomach acid. And with weakened defenses, the stomach acid and the pepsin, which is the protein digesting enzyme in the stomach, are able to start eroding the stomach lining, which can lead to a peptic ulcer. So they can actually get down here and start breaking apart the cells that make up the mucosa. Number 5, why is the stomach so acidic? Describe how the acid is produced and secreted and the 4 functions for the acidity. So the stomach is acidic because parietal cells release hydrochloric acid into the stomach lumen. The acid is secreted into the lumen as separate hydrogen ions and chloride ions as follows. So carbon dioxide is first combined with water to form carbonic acid, which is, this whole reaction is catalyzed by the enzyme called carbonic anhydrase. The carbonic acid, so the H2CO3, then dissociates into hydrogen ions and bicarbonate ions. So remember, this is an equation that's going to keep coming up for the remainder of the semester. So carbon dioxide joins together with water to form carbonic acid, H2CO3, carbonic acid then dissociates into hydrogen ions and bicarbonate ions. So the hydrogen ions are then pumped into the lumen of the stomach using these proteins called proton pumps. And meanwhile, the bicarbonate acid that was formed is being swapped for a chloride ion from outside of the epithelial cell. So the bicarbonate ion is leaving the epithelial cell and a chloride ion is coming in, and that is called the chloride shift. And then that chloride ion is then pumped out into the gastric lumen. Now we have hydrogen ions out here and chloride ions out here, so now we have hydrochloric acid in the lumen of the stomach. So the 4 functions for stomach acidity are to kill ingested microorganisms, to denature proteins and inactivate most enzymes, to help break down plant cell walls and connective tissues in meats, and to allow pepsinogen to be converted into the active enzyme pepsin, which starts the chemical digestion of protein. Number 6, what does the coffee ground appearance of the blood indicate? So as discussed earlier, when blood spends a significant time in the acidic environment of the stomach, the acid oxidizes the iron in the hemoglobin, turning it a dark brownish black color. Pepsin also begins to digest the hemoglobin protein, causing the blood to break up into small specks. Thus, the blood takes on the appearance of coffee grounds. So the most common reason for coffee ground vomitus is a peptic ulcer, either in the stomach or the duodenum. So a peptic ulcer is going to be when the acid and enzymes of the stomach eat through the mucosa layer and possibly even down into the submucosa, and this can happen either inside the stomach itself or inside the duodenum where the stomach acid goes next. Note that if a person never vomits while they have a peptic ulcer, the blood can pass all the way through the digestive tract and result in black tarry stools. So the black color and the strong odor are characteristic of hemoglobin digestion by the digestive enzymes in the digestive tract and the intestinal bacteria in the large intestine. Number 7, what do you think reflux esophagitis means? What things could cause this to happen, and what normally prevents this from occurring? So reflux esophagitis is the inflammation of the esophagus due to acid reflux from stomach acid. So normally you have the lower esophageal sphincter that closes to keep the acidic chyme that is in the stomach from coming back up into the esophagus. You also typically have high alkaline mucus production by the cardia region of the stomach. And with acid reflux, you have the sphincter becomes weakened and the stomach acid can go back up into the esophagus. The esophagus is not built to deal with the acid contents, so the acid can start to irritate and erode away the lining of the esophagus. So to answer the question about what normally prevents this from occurring, so normally the lower esophageal sphincter prevents the acidic chyme from moving back up into the esophagus, and the cardia region of the stomach produces excess alkaline mucus to help protect the esophagus. So alcohol is known to relax the lower esophageal sphincter, which will more easily allow acid from the stomach to move back up into the esophagus, irritating the lining of the esophagus and causing inflammation and pain, which is esophagitis. Other causes of acid reflux can include excessive intake of caffeine or chocolate, both of which also loosen the lower esophageal sphincter. Smoking relaxes the lower esophageal sphincter as well. Eating very large meals, like at an all-you-can-eat buffet, can cause over-distension of the stomach, and so the pressure of all of that food inside the stomach can push back and cause the chyme to leak back up into the esophagus. And then lying down within 2 hours of eating a large meal puts pressure on the lower esophageal sphincter, because when you're lying down, you no longer have gravity that is pulling the chyme to the bottom of the stomach. So the chyme is more likely to go back upwards and put pressure on that lower esophageal sphincter. And then age. So all muscles, including your sphincters, wear out as you age. And acid reflux is the most common cause of heartburn, and it got the name heartburn because if you remember, the esophagus passes behind your heart, and so it can feel like pain in your chest, but it's actually your esophagus. So we're still focusing on poor Vincent, and now we are looking at hospitalization number 3. So at age 41, Vincent entered the hospital with complaints of a high fever, nausea, loss of appetite, and a dull continual pain in the left side of his back. In addition, he had diarrhea of a particularly foul odor and yellow color. He had also lost 15 pounds over the last month and a half. So number 8, describe the organization of membranes, mesenteries, in the digestive system. What are the functions of the mesenteries? So the mesenteries are a double sheet of peritoneal membranes that are connecting the parietal and visceral peritoneum. So as you can see in this image, the mesentery is going to be a double sheet of peritoneal membranes. And to remind you, the parietal peritoneum is lining the peritoneal cavity wall, and the visceral peritoneum is lining the organs inside the peritoneal cavity. So the mesentery is the double sheet of membrane connecting the two. So if we look at the different mesenteries, we have the falciform ligament that connects the liver to the abdominal wall or diaphragm. There is the lesser omentum shown in purple, which is connecting the liver to the stomach. There is the greater omentum, which connects the stomach to the transverse colon, and it folds down to form like an apron or a pouch. So it folds down here in the front of the abdomen. There is the mesentery proper, which is shown with the green outline. This connects the loops of the small intestine. Then there is the mesocolon, which stabilizes the large intestine, and that is shown, a portion of it is shown up here with a brown outline. So the mesenteries function to provide a route for blood vessels, nerves, and lymphatic vessels to get to and from the organs in the peritoneal cavity. They also stabilize the position of the attached organs, and they prevent entangling of the intestines during digestive movements or sudden changes in body position. So number 9, inflammation of which digestive organ is likely causing Vincent's back pain? Hint, based upon the location of this pain, would you guess that the organ in question is a retroperitoneal organ or an organ attached to the abdominal wall by a broad sheet of mesentery? So pain felt in the back usually involves a retroperitoneal organ, which is an organ that sits behind the parietal peritoneal membrane. So if you look at this picture here, it will show you the peritoneal cavity is shown in this purple area. And so the peritoneal cavity would be lined with the parietal peritoneum. So anything that sits behind that membrane would be considered to be retroperitoneal. So retroperitoneal organs, you can remember them by using the mnemonic SAD pucker. The S stands for the suprarenal gland, which is the same thing as the adrenal gland, but if you call it the suprarenal gland, which means it sits on top of the kidney, then you can use the S. A stands for the aorta and the inferior vena cava. D is the duodenum, half of it. P is the pancreas, U is the ureters, C is the colon, K is the kidneys, E is the esophagus, and R is the rectum. So parts of the digestive system that are retroperitoneal are going to include half of the duodenum and the pancreas, the ascending and descending colon, and the esophagus and the rectum. So based on Vincent's weight loss and diarrhea, which we're going to explain in an upcoming question, Vincent most likely has alcohol-induced pancreatitis. Number 10, describe the anatomy of the organ identified in question 9. So the pancreas sits just underneath the stomach with its head resting in the curve of the duodenum. It is covered by a thin, transparent capsule of connective tissue, which also partitions the pancreas into distinct lobules, and they're showing you one of the lobules in this image. 99% of the pancreatic cells are exocrine acinar cells. The other 1% are endocrine islet cells. The pancreatic acini secrete digestive enzymes and bicarbonate buffers into small ducts, which then combine to form the large pancreatic duct. And the pancreatic duct releases the secretions into the duodenum at the duodenal ampulla, which is controlled by the hepatopancreatic sphincter. So in the case of alcohol-induced pancreatitis, so alcohol gets converted into toxins, so alcohol is a poison, and so it can cause all sorts of things to happen inside of the pancreas. You can get some abnormal blood flow and secretion, some toxic metabolites, some oxidative stresses. The details aren't important. The take-home message is that chronic use of alcohol can end up causing inflammation of the pancreas, which is going to lead to pancreatitis. So number 11, based upon the function of the organ identified in question 9, what is causing the diarrhea and weight loss? So the pancreas is responsible for secreting the majority of the digestive enzymes, which include pancreatic alpha amylase, pancreatic lipase, nucleases, and a whole bunch of proteolytic enzymes like trypsin. So with pancreatitis, the amount of secretions is greatly reduced, and so since you have fewer enzymes being secreted, chemical digestion inside the small intestine becomes impaired. So without chemical digestion to break the large macromolecules down into their smallest subunits for absorption, the food ends up passing through the digestive tract unabsorbed. This is going to cause weight loss because the nutrients are not getting absorbed into the body from the digestive tract. So it's kind of the same thing as not eating at all. So this would explain his sudden and rapid weight loss. Since the food is not being absorbed and is staying in the digestive tract, this also throws off the osmotic balance of water movement. So let's take a look at that. So normally when food enters the small intestines, you have a lot of solute inside the lumen and not much of it has been absorbed yet. So you have a lot of water flowing into the intestine. Remember, water moves toward areas of high solute. But then as the food passes through the small intestine and into the large intestine, you've got the nutrients are being absorbed into the body. And so that is reducing the solute concentration inside the digestive tract, so that as you move down and the solute concentration becomes less and less, water goes back into the body. So it's reabsorbed because, again, water is moving towards high solute. And now when you get down to the end of the large intestines, you have low solute inside the intestines. So water is moving back out into the body, so it gets reabsorbed. So we lose very little water normally in our fecal matter. Now let's see what happens in pancreatitis. So in pancreatitis, remember, we're not getting chemical digestion because we're not secreting the digestive enzymes from the pancreas. So without chemical digestion, we're not getting nutrient absorption, right? So we're not breaking these things down into small enough pieces that they can be absorbed across the digestive epithelium. And so remember, that was what caused the weight loss, is because he's not being able to absorb any nutrients. But that also means that the solute concentration inside the digestive tract stays high all the way through because the solutes are not being absorbed. And so water's going to come in at the beginning, but then there's going to be no driving force for the water to be reabsorbed later on, because you still have a high solute concentration inside the intestines when you get to the end, because you haven't absorbed anything. So water stays in the digestive tract and is not reabsorbed. And so that results in a lot of water in the stool, which gives you a very watery stool, which is diarrhea. The undigested lipids and fats, again, nothing is being digested, so nothing can be absorbed, is what is responsible for giving the stool a yellow color and a foul odor. And did you know there is also a disorder called the exocrine pancreatic insufficiency, or ESI, in which the pancreas doesn't produce enzymes normally, and you would have the same symptoms of diarrhea and weight loss. Okay, so we're still on the case with Vincent, and now we're going to look at hospitalization number 4. Hospitalization 4. Vincent was then hospitalized again at age 49 with dull pain in the right upper quadrant of the abdomen, intermittent fever of 3 weeks duration, and a yellowing of the skin and the whites of the eyes. Number 12, what organ is involved this time? Describe the anatomy of this organ and blood flow through this organ. So Vincent is now suffering from alcohol-induced hepatitis, which is inflammation of the liver caused by excessive alcohol consumption. So the liver contains 4 major lobes. So from the anterior view, you can see the right lobe and the left lobe. And then from the posterior view, you can see the caudate lobe and the quadrate lobe. The porta hepatis is shown here. This is the area where the hepatic artery proper and the hepatic portal vein take blood into the liver, and the common hepatic duct takes bile away from the liver. The liver contains approximately 100,000 lobules, with each lobule being separated from the others by an interlobular septum. And so here is a different view from your textbook showing you the liver lobule. So each lobule is roughly hexagonal, meaning it has 6 sides, and at each of the 6 corners it has a portal triad, which is a branch of the hepatic artery, a branch of the hepatic portal vein, and a bile duct. Sinusoid capillaries run through 1-cell thick plates of hepatocytes, shown in this image as well, and on their way to the central vein, which is located in the middle of the lobule. So now we're going to look at the blood flow. So oxygenated blood comes into the liver through the hepatic artery proper. It then branches into smaller and smaller branches until you get an interlobular artery that dumps oxygenated blood into a sinusoidal capillary at the portal triad. So up here at the top, we're looking at one portal triad where we have a branch of the hepatic artery, a branch of the hepatic portal vein, and a branch of the bile duct. We see the central vein here in the middle. So oxygenated blood is coming in through the hepatic artery branch and is then joining up with the sinusoidal capillary, and you can see all of this in this bottom picture as well. And the bottom picture shows you that blood flow is going from the outside inward to the central vein, whereas bile flow is going in the opposite direction. So deoxygenated blood that is leaving the intestines through the mesenteric veins enters through the hepatic portal vein system, and the hepatic portal vein also branches into smaller and smaller branches until eventually you get an interlobular vein that dumps the blood also into the sinusoidal capillary at a portal triad. So blood is entering through the artery and the vein at the portal triad into the sinusoidal capillaries. So once blood from both sources enter the sinusoidal capillaries, the blood moves between the hepatocytes to the central vein in the middle. All of the central veins will connect up to form the hepatic veins, which you can see in the back of this image. There's the three little hepatic veins here, and these hepatic veins leave the liver and join up with the inferior vena cava. So again, remember the hepatic portal vein and the hepatic veins are different vessels. So let's look at the blood flow, little graph here. So from the systemic veins that are leaving the intestines, so through the inferior and superior mesenteric veins, blood is going to go into the hepatic portal vein. It will then go into the liver, where it will branch into smaller and smaller branches and eventually form the interlobular veins at the portal triads. It will then go into the sinusoidal capillaries. Meanwhile, we also have oxygen-rich blood from systemic arteries coming into the liver through the hepatic artery proper, which also branches into smaller and smaller branches inside the liver until it gets to an interlobular artery at one of the portal triads, and then this blood is also going to be dumped into the sinusoidal capillaries. So all of this blood is then going to flow through the liver lobule to the middle central vein. Then after all of the central veins come together, they will form the hepatic veins that leave the liver, which will take the blood to the inferior vena cava, where it will be returned to the heart, the right atrium specifically. Number 13, why are Vincent's skin and eyes tinged yellow? What is this condition called? Review the production and flow of bile, and describe the structure and function of bile salts. So the yellow color in the skin and eyes comes from an increased level of bilirubin in the blood. So if you remember from earlier chapters, bilirubin comes from the breakdown of the heme group of hemoglobin during red blood cell recycling. So the condition of having elevated levels of bilirubin in the blood, which causes the yellowing of the skin and the eyes, is called jaundice. So normally, the liver filters the bilirubin out of the blood and secretes it in bile. But hepatitis can prevent the hepatocytes from properly filtering out the bilirubin, which can cause excess bilirubin to build up in the bloodstream. So the hepatocytes of the liver are the ones that produce the bile, and bile is composed of water, ions, bilirubin, cholesterol, and bile salts. Bile salts are synthesized from cholesterol. They are actually part hydrophobic and part hydrophilic. So if you look at a single bile salt molecule, part of it has a water-fearing side and part of it has a water-loving side, and this allows them to surround the lipids. So their hydrophobic side faces the lipids, the hydrophilic side would face the aqueous environment inside the digestive tract. So the function of the bile salt is to emulsify large lipid droplets into much smaller lipid droplets so that the lipase enzymes can get in and break the bonds. So emulsification is the process of taking a large fat droplet and bile salts, and again the bile salts will surround the lipids because of their region of hydrophobicity, and so it's going to break them down into very small fat droplets. So you go from a small surface area to volume ratio to a very large surface area to volume ratio. The bile salts are then reabsorbed by the ileum of the small intestine and then recycled by the liver from the hepatic portal circulation. So to continue with this question, let's look at the bile flow, release, and storage. So the hepatocytes in the liver are making the bile, and after the hepatocytes make the bile, it is secreted into the bile canaliculi within the liver lobule. And then the bile is going to flow outward in that lobule to the interlobular bile duct that is at the portal triad. So remember, that's at the corners, the 6 corners of each liver lobule. All of the interlobular bile ducts will come together to form the left and right hepatic ducts. The left and right hepatic duct will join together to form the common hepatic duct, which leaves the liver through the portal hepatis. Bile can then flow through the cystic duct and into the gallbladder for storage, but it can also flow into the common bile duct where it will enter the duodenum through the duodenal ampulla, which is controlled by the hepatopancreatic sphincter, and that will allow for the release of bile into the small intestine. Also notice that you can also take bile that was previously stored in the gallbladder and move it back out through the cystic duct and then through the common bile duct and into the duodenum for release. So number 14, if Vincent's disorder resulted in the production of a gallstone, what impact might that have on Vincent's pancreas? So there are 2 main types of gallstones that can form. Some of them can form from cholesterol, and this is the most common type. And interestingly, whether or not you have bile stones that are formed from cholesterol is actually not related to your blood cholesterol levels. So even if you have normal blood cholesterol levels, you can still form gallstones. But you can also have stones that are formed from bilirubin, and these can result from excess bilirubin, like if you have a condition that causes you to have an abnormally high red blood cell turnover. So if gallstones block the cystic duct or the common bile duct, then it can cause pain, inflammation, and potential jaundice from the backup of the bilirubin excretion. If a gallstone actually lodges in the duodenal ampulla, though, it's going to block not only bile flow, but it will also block secretions from the pancreas. So remember, both the pancreatic duct and the bile duct empty into the duodenal ampulla. So a gallstone here would block secretions from both organs. And then the digestive enzymes produced by the pancreas could get backed up and start digesting the pancreas itself, which could lead to inflammation, which could potentially give Vincent yet another round of pancreatitis. So number 15, describe the hormonal control of the secretion of bile and pancreatic juice during the digestive process. So note that this question is only asking about the hormones that regulate bile and pancreatic juice, which is the enzymes and buffers made by the pancreas. So it's not asking about all of the duodenal hormones, just the ones that regulate these 2 fluids. So here's a picture. At the top we can see the regulation of the pancreatic juice. And again, pancreatic juice is made up of the buffers, which contain mostly bicarbonate, and digestive enzymes. And then down here we can also see the pancreas, the buffers, and the enzymes, but we can also see the gallbladder and the production of bile by the liver. So secretin is released by the duodenum when chyme first enters it. And secretin is going to stimulate the production of alkaline buffers by the pancreas. All right, so it's shown here, secretin is going to stimulate the bicarbonate buffers. It's also shown down here, secretin stimulates bicarbonate buffers. Secretin also stimulates the hepatocytes to produce and secrete bile. So it's telling the liver to make more bile. Then we have cholecystokinin, or CCK, which is also released by the duodenum when chyme enters. CCK is going to stimulate the production and release of the digestive enzymes from the pancreas. So remember, pancreatic juice has two components, the buffers and the enzymes. And again, down here, we're showing that CCK is going to stimulate the production and release of the enzymes. CCK is also going to stimulate the gallbladder to contract, which will release stored bile. And not shown in this picture, but CCK will also relax the hepatopancreatic sphincter so that both bile and pancreatic juice can enter the duodenum. Now note that the gastric inhibitory peptide, or GIP, can also stimulate the pancreas to produce insulin, but insulin is released directly into the bloodstream and is not part of what we call pancreatic juice. The pancreatic juice is only the exocrine secretions of the pancreas, and it only includes the digestive enzymes and the buffers. Number 16, what long-term effects could Vincent's alcohol consumption have on his liver? So there are two main ways that alcohol adversely affects the liver. First, alcohol can cause hepatitis, and chronic hepatitis can lead to cirrhosis. Secondly, because of the amount of carbohydrates in alcoholic beverages, the liver stops processing fatty acids for energy and instead stores those fatty acids within the liver, which can lead to a fatty liver. You can also get a fatty liver from just consuming a lot of sugar in your diet. In fatty liver disease, the excess fat deposits start interfering with normal liver function and can cause inflammation, which again can lead to hepatitis. In both cases, the chronic inflammation of the liver will eventually result in the development of scar tissue within the liver, which is called fibrosis. And the scar tissue will take the place of the normal hepatocytes, which reduces the overall liver function. So cirrhosis of the liver occurs when a significant portion of the liver has become replaced by fibrous scar tissue. And this is, at this stage, it is permanent damage to the liver and cannot be undone. So the green arrows in this image show the two stages at which you can reverse some of the damage by stopping your alcohol consumption. But once you've passed one of these red arrow points, then there's no going back. So cirrhosis will also increase your chances of getting liver cancer, which is called a hepatocellular carcinoma. So conclusions from Vincent's case study. Hopefully by now you see that alcohol is not very good on your digestive system. It can increase the likelihood of heartburn, compromise your stomach lining, increase your risk of pancreatitis, interfere with your nutrient absorption, and then we didn't even talk about this, but can also cause imbalances in your large intestine gut bacteria. And they didn't mention the cirrhosis, so I put another picture up here that shows the cirrhosis to the liver. And then, of course, alcohol also can affect other systems. It can cause kidney failure, it can cause problems with the heart, and it can cause damage to brain areas. Basically, alcohol is a poison, so it shouldn't be surprising that it has this widespread of damage on the body's systems. So now we're done with Vincent, and we'll move on to part 2, some miscellaneous questions. So number 1, imagine eating a sausage biscuit and a piece of fruit for breakfast. What major macromolecules are found in the sausage biscuits and fruit? Describe the digestion of this food item as it passes through the digestive system. So a sausage biscuit and the fruit would contain all of the major macromolecules. So you would have protein, carbohydrates, and lipids or fats. So let's start in the oral cavity. So you put the food in your mouth and you start chewing it. So in the oral cavity, your teeth and your tongue would participate in mechanical digestion, which is the physical breakdown of the food into smaller pieces. So no chemical bonds are broken, but the food is broken down into smaller particles to increase the surface area for the enzymes to come in and break bonds. Carbohydrate chemical digestion would begin in the oral cavity with salivary amylase that is secreted by the salivary glands, and amylase breaks down starch, which is a type of carbohydrate. Lipid chemical digestion would also begin in the oral cavity with lingual lipase, which is secreted by the tongue. Food would then pass into the pharynx and the esophagus. So food would pass into the pharynx and then the esophagus during deglutition, which is swallowing, and then it would travel to the stomach. So now we're in the stomach. We would have additional mechanical processing occur here. So there would be churning, segmentation, which is helped along by the third layer of oblique muscle in the stomach wall. You would also have your parietal cells releasing hydrochloric acid, and the acid would help to break down the connective tissues in the meat and the plant fibers in the fruits. As the pH drops below 4.5 because of the production of the acid, at this point the salivary amylase and the lingual lipase from the oral cavity are inactivated and broken down themselves. And then the chief cells are also secreting pepsinogen, and as the pH falls to around 2, pepsinogen is converted to pepsin, and chemical digestion of the proteins will begin. So then the stomach contents, which is called chyme, will move into the small intestine. As the chyme from the stomach enters the duodenum, bile from the liver is added to the mix, as well as pancreatic juice from the pancreas. So remember, the duodenum is a mixing bowl that mixes the chyme from the stomach with secretions from the liver and the pancreas. So the bile salts in the bile from the liver help to emulsify large lipid globs into tiny lipid droplets with more surface area. Pancreatic enzymes will continue to break down chemical bonds in the process of chemical digestion. So some of the ones we talked about in lecture were alpha amylase, which breaks down starch, which is a type of carbohydrate. We also talked about lipase, which breaks down lipids, nucleases, which break down DNA and RNA, and then there were a lot of proteolytic enzymes we talked about, like trypsin, chymotrypsin, carboxypeptidase, and elastase, which break down proteins. So continuing with the small intestine, so as those enzymes were breaking the large macromolecules down into smaller pieces, eventually with the carbohydrates you get down to disaccharides and trisaccharides, and then they are broken down by brush border enzymes on the microvilli of the intestinal cells to monosaccharides, and monosaccharides is the smallest unit. They are absorbed by facilitated diffusion and then pass into the bloodstream. Likewise, eventually the proteins are broken down into little pieces of 2 amino acids called dipeptides. The dipeptides are then broken down by brush border enzymes to single amino acids, and amino acids are then absorbed by facilitated diffusion and also pass into the bloodstream. Fatty acid tails and monoglycerides that came from the breakdown of lipids are absorbed into the intestinal cells by diffusion. Once inside the intestinal cell, they are reassembled into triglycerides and packed with proteins to form chylomicrons. And chylomicrons are secreted by the intestinal cell using exocytosis into a lacteal, which is a lymphatic vessel. So fats will enter the bloodstream indirectly by taking a little detour through the lymphatic system first. So now we have done the bulk of our absorption and our digestion. So now what is left moves into the large intestine. So in the large intestine, at this point, we mainly have just indigestible matter left over, like plant fiber. Bacteria in the large intestine can use this indigested material as a food source, and then they produce vitamins that we can absorb, like vitamin K, biotin, and pantothenic acid. Final water absorption will also take place in the large intestine. And then whatever is left after all of this, the final waste material is then compacted and eventually removed from the body by defecation. Number 2, what are the 6 integrated functions of the digestive system? Why are they called integrated functions? So first, to do the 6 functions. 1 is ingestion, and this is when food is entering the oral cavity when you eat or drink something. Then we have mechanical digestion or mechanical processing. This is crushing, shearing, and the use of other mechanical forces to physically break the food down into smaller pieces to increase the available surface area. So we're not breaking chemical bonds. This is a physical change. So for example, like when you chew something up, you're breaking it into smaller pieces. Then we have chemical digestion, which is actual breaking the chemical bonds between the smaller subunits that make up these large macromolecules. Then we have secretion. So as you've seen by now, the digestive system releases a lot of water, acids, enzymes, buffers, and salts into the lumen. So some examples that we've talked about along the way are saliva, gastric juice, pancreatic juice, bile, intestinal secretions, and mucus. So that is the function of secretion. Absorption is the movement of the smallest organic subunits, so like the monosaccharides, the fatty acid tails, the monoglycerides, and the amino acids, across the digestive epithelium and into the body's fluids. And then defecation or excretion is removal of the waste products from the body. So the reason these are called integrated functions is because each function depends on one or more of the other functions, with the exception of ingestion. So to eat something to start with doesn't really depend on everything else in this list, but everything else in this list is going to be dependent on at least one other thing. So for example, if you have not mechanically processed the food to increase its surface area, then chemical digestion would not occur efficiently because the enzymes couldn't get into the bonds that they need to break. Another example, if you don't have any of the secretions like pancreatic juice, you won't get the enzymes that are necessary for the chemical digestion to occur. And to give you another example, if you don't chemically digest the food into its smallest subunits, then absorption will not occur. And then, of course, like if you don't eat anything at all, you won't have anything to defecate or excrete. So all of these functions are sort of interreliant on each other, and that's why they're called integrated. Number 3, describe the 3 phases of gastric regulation. So the first one is the cephalic phase. In this phase, the sight, smell, taste, or thought of food causes the central nervous system and the enteric nervous system to activate mucus cells, chief cells, parietal cells, and G cells in the stomach. So the mucus cells are going to produce protective alkaline mucus. The chief cells will produce pepsinogen. The parietal cells produce hydrochloric acid. And the G cells produce the hormone gastrin, which will further increase the production of these other substances. The next phase is the gastric phase, which starts with the arrival of food into the stomach. And so this phase is sensed by stretch receptors that sense the distension of the stomach. Also, there are chemoreceptors that can sense an increase in pH and the presence of undigested proteins. So in the gastric phase, you have further activation of the mucus cells, chief cells, and parietal cells. And now you also trigger the mixing waves or segmentation in the muscular layer to help to start churn the stuff in the stomach, and that is part of the mechanical processing. Then you have the intestinal phase, which starts with the arrival of chyme into the duodenum, and this is sensed by stretch receptors that can sense the duodenum distend, and also chemoreceptors that can sense a decrease in pH in the duodenum. So the intestinal phase is going to involve the pyloric sphincter contracting or closing off, which will limit the amount of chyme that is entering the small intestine from the stomach, and this is called the enterogastric reflex. The stomach secretions are reduced, and the stomach then becomes a holding tank for the food until the duodenum is ready to handle more chyme. So remember, the duodenum has to mix the chyme with bile and pancreatic juice, so it only wants to handle a little bit at a time. So basically, as soon as some enters the duodenum, the goal of all of this is to shut the stomach down or slow it down so that you have time to take care of what is currently in the duodenum before adding more. So some of the hormones that get released are gastric inhibitory peptide, or GIP, which decreases the motility of the stomach and the stomach secretions. Secretin also decreases the motility of the stomach and the stomach secretions. Cholecystokinin, or CCK, decreases the motility of the stomach, which slows the rate of emptying into the duodenum. And the vasoactive intestinal peptide, or VIP, reduces acid production in the stomach, which helps to protect the stomach while it serves as a holding tank waiting on the duodenum to be ready for its next bout of chyme. Number 4, describe the anatomy of the large intestines. So histologically, there are no villi, but there is an abundance of mucus cells called goblet cells. There are deeper intestinal glands, and there are also large lymphoid nodules, which is part of the malt in the lamina propria of the large intestine. The large intestine anatomically can be broken into 3 main sections. The first one is the cecum, shown here. The cecum is an expanded pouch at the area where the ileum joins the large intestine, and this is where you also have the ileocecal valve. Food entering the stomach will actually trigger the opening of the ileocecal valve, and that is called the gastroileal reflex. The appendix protrudes from the cecum, and the appendix is also malt, which is mucosa-associated lymphoid tissue. So that is part of the lymphatic system. The next part of the large intestine is the colon. So this is the main part of the large intestine. It has these little pouches called haustra. So haustrum, as shown here, would be singular. And there is also this band of muscle that runs along it called the tenei coli. The colon can be subdivided into 4 segments. So starting at the cecum, you go up through the ascending colon, then you go across through the transverse colon, then down through the descending colon, and then through the little curved part called the sigmoid colon. And the final part of the large intestine is the rectum, which is an expandable pouch that makes up the last 6 inches of the digestive tract. This allows for the temporary storage of feces prior to elimination. And the movement of material into the rectum is what triggers the urge to defecate. The rectum has both internal and external anal sphincters. In this picture, you can only see the external anal sphincter. But the external anal sphincter is under our voluntary control until you wait too long, and once the pressure inside the rectum builds up to 55 millimeters mercury, it is no longer voluntary and becomes involuntary. And the last question, number 5, describe the structures associated with the oral cavity. So the gingivae, also called the gums, are the ridges of oral mucosa that surround the teeth. And the gingival sulcus is that shallow groove around the neck of each tooth where the mucosa epithelium is actually bound to the tooth. Another structure is the hard palate, which is the bony roof of the mouth made up of the maxilla and palatine bones. The hard palate has ridges that give traction for the mechanical processing of food in the oral cavity. The soft palate is at the border between the nasopharynx and oropharynx, so it sits behind the bony portion of the palate. The soft palate can fold back to seal the nasopharynx off during swallowing or vomiting. And the uvula is this little small extension down on the end of the soft palate, which helps to prevent food from entering the pharynx too soon. The tongue is a large muscle, and on the superior surface it is covered with taste buds, which contain gustatory receptors, as we talked about earlier this semester. The tongue helps with mechanical processing of the food. It also secretes mucins and lingual lipase. It helps to manipulate the food to assist in chewing and to prepare food for swallowing. And it provides sensory analysis, so touch, temperature, and taste of whatever is in your mouth. The next structure are the salivary glands, and there are 3 pairs of glands that work together to produce saliva. There are the parotid glands, which secrete a large amount of salivary amylase. There are the sublingual glands, which secrete buffers and lubricant. And there is the submandibular glands, which secrete buffers, mucins, and salivary amylase. Then we have our teeth, which are responsible for mastication or chewing. The crown or top part of the tooth is made up of enamel, but the bulk of the tooth is made up of a substance called dentin, which is very similar to bone, but it has no cells. The tooth is anchored to the bone by the periodontal ligament and a layer of cement, which can also be called cementum, as shown in this picture. And the joint where the tooth sits in the bone of the upper or lower jaw is called a gomphosis. The pulp cavity is deep in the inside of the tooth, and this is where the blood vessels and nerves are located, and they enter the tooth through this channel called the root canal. And an adult has 32 permanent teeth. They have 4 incisors on top and the bottom, so 8 total. 2 canines or cuspids on the top and bottom for 4 total. 4 premolars or bicuspids on the top and bottom for 8 total. And 6 molars on the top and bottom for 12 total, and that does include the wisdom teeth. And then that is it for the Chapter 24 activity.